Hello students, welcome back to my biology online classes. Today I am going to discuss with you about previous year question paper named Genetics under Cbcs 5th semester. This is a question from University of Calcutta year 2023 for your upcoming semester 3 genetics exam under CCF. Now let's concentrate. Number 1. Distinguish between complete and incomplete P elements in Drosophila carrying 2 marks. Define extra chromosomal hereditary factors carrying 2 marks. What is intercalary deficiency? Give example from Drosophila carrying 2 marks. Distinguish between composite and non-composite transposition carrying 2 marks. Define the terms dominant epistasis and duplicate epistasis carrying 2 marks. What is gynandromorph carrying 2 marks? Define isochromosome carrying 2 marks. Distinguish between amorphic and hypomorphic mutation carrying 2 marks. Define with example numerator and denominator elements in Drosophila sex determination carrying 2 plus 2 marks. How X is to A ratio determines embryonic and adult functional asexual production in Drosophila carrying 4 marks. Define pleiotropic gene effect carrying 2 marks. Describe how studies using the Drosophila lozenge locus revealed that recombination could occur within a gene carrying 5 marks. Define prototroph and auxotroph in Neurospora sp carrying 2 marks. Explain position effect of translocation carrying 2 marks. Give an example of terminal translocation in human carrying 1 marks. Describe ACDS element in maize carrying 3 marks. Plumage color in mallard ducks is dependent upon a set of 3 alleles. MR for restricted mallard pattern, capital M for mallard and small m for dusky mallard. The dominance hierarchy is MR is greater than capital M is greater than small m. Determine the genotypic and phenotypic ratio expected in F1 from a cross. Number 1, MR capital M into capital M small m and MR capital M into small m carrying 2 plus 2 marks. Next question. A non-hemophilic woman whose father was hemophilic was paired with a non-hemophilic man. Now the problem is number 1. What is the probability that they will have a hemophilic male child carrying 1 mark? What is the probability that they will have a hemophilic daughter carrying 1 mark? What is the genotype of the offspring? 1 mark. Next question. In Drosophila, the male X chromosome transcription is upregulated approximately 2-fold in somatic tissues. Explain, including epigenetics of the control carrying 5 marks. Next question. In corn, the kernel color may be white, purple or red and is controlled by 2 genes I and P. Corn that is I underscore P underscore is red. Corn that is I underscore PP is purple and corn that is small I P underscore or small I small p is white. A dihybrid cross shows a ratio 9 red is to 3 purple is to 4 white. Explain the gene interaction involved carrying 3 marks. Write two important applications of transposable genetic element carrying 2 marks. Explain with example the process of mutation in DNA by base analog and UV light carrying 2 plus 2 marks. Design a cross to explain primary non-disjunction in Drosophila. State the inference drawn from the result carrying 5 plus 1 mark. Inversions are often called crossover suppressors. Explain carrying 2 marks. Describe the role of SRY, SOX9 and DAX1 in human sex determination carrying 2 plus 2 plus 2 marks. How a mosaic XX/X0 human can be formed carrying 2 marks. Next question, a cross in Drosophila involved the recessive X-linked genes yellow, small y, white, small w and cut, CT. A yellow-bodied, white-eyed female with normal wings was crossed to a male whose eyes and body were normal but whose wings were cut. The F1 females were wild type for all 3 traits, while the F1 males expressed the yellow body and white eye traits. The cross was carried to an F2 progeny and only male offspring were tallied. On the basis of the data shown here, a genetic map was constructed. Look carefully. In left hand side phenotype and right hand side male offspring, Y plus CT 9 plus W plus 6, YWCT 90 plus 95 plus CT 424, YW plus 376, Y plus 0 and plus WCT 0. From this genetic map, 4 problems. Number 1, determine the genotypes of the F1 parents carrying 2 marks. Find correct gene order carrying 2 marks. Construct a map assuming that white gene is at locus 1.5 cm on the X chromosome carrying 4 marks. Find CC and interference carrying 2 marks. So students, this is all about previous year genetics paper of University of Calcutta under CBCS 5th SEM for your upcoming semester 3 exam under CCF regulation. Stay connected for next session of previous year question discussion.